Hi everyone, this is Mrs. Bustamante, and today we're going to be talking about 6.4, which is the process of translation. Basically, it's the big second step in the order of what we call protein synthesis, right? How do we go from DNA to proteins? And what are all the steps there, and what should you be able to do with it? So where we left off, right, we were talking last time in our videos about the process of transcription. And we were also talking about the idea of central dogma, right, that we go from DNA to RNA to protein. Well, and to go from DNA to RNA, we said the first step to do that is something called transcription, right? We change it from the DNA language into the RNA language. Now we're going to talk about the second step, which really translates it from that RNA language into our protein, or more specifically, kind of just our polypeptide chain, but we know that will fold and probably even combine with some other proteins to become a functional protein. So we're really talking about the second step of protein synthesis in this video. What is translation then, right? Well, it's really just the process by which an mRNA sequence that was generated from our DNA, right? We used our template strand to come up with that mRNA strand. We processed it if it was a eukaryotic cell, right? And then it left the nucleus, again, if it's eukaryotic cell. And now it's going to go to this ribosome and it's going to get translated. Okay, this is kind of the last step in the flow of our information. It occurs at the ribosomes. In prokaryotic and eukaryotic cells. However, prokaryotic cells only have ribosomes in the cytosol. So that's where it's going to occur. In eukaryotes, right, we have ribosomes that are free in the cytosol, but we also have ribosomes that are bound to the rough endoplasmic reticulum, and it will occur in both of those locations in eukaryotic cells. So we're really talking about the second half of the process here. Here are some terms you're going to want to know and understand, and again, be able to use and explain pretty freely. Some of them will seem kind of like repeats from transcription because we're putting those together, and really a lot of things by now you have heard a lot about, so it should be familiar. So what are our steps of translation? We're really going to talk about translation in 3 steps. We're going to have an initiation step, aka a starting step, an elongation step where we make that polypeptide chain longer, and then a termination step, basically where we end it. In prokaryotes, it's important to note, remember we don't have a nucleus, right? So translation occurs basically at the same time as transcription. Where in eukaryotic cells, it's a little more sequentially stepped, right? Because we have to have that mRNA leave the nucleus before it can go onto the ribosome and be translated. So what's our first step? Our first step, right, is initiation. We just said that. The rRNA part of the ribosome is going to interact with the mRNA that has left the nucleus. And it's going to connect, if you will, at what we call the start codon. In a little bit, we're going to talk about how the start codon is a specific part of the mRNA. It will always be the sequence of AUG nucleotides. We'll talk about why that is important in a little bit. mRNA contains nucleotides, right? We're going to group them into 3s, okay? So we're going to call this a triplet. And one triplet, rather 3 nucleotides together, is equal to 1 codon. And 1 codon eventually will code for 1 amino acid. I'm going to say this again. mRNA contains the codons. I'm going to say that a lot because it's really, really important. So each codon on your mRNA molecule will code, see the connection there, for a specific amino acid. Eventually we're going to use a codon chart to determine what that is, but just hang on to that a little bit for a second, okay? So what do I want you to look at here? Here's my ribosome. Ribosomes actually have 2 parts. And so we call them a large subunit and a small subunit. Really, our small subunit is going to kind of be our binding site, I guess, if you will, for our mRNA sequence. And our mRNA sequence is going to go to this ribosome. It's going to kind of hold it in place. We're also going to talk about the P site, the A site, the P site, and the E site, if you will. because those sites are really important. It's going to kind of move like a conveyor belt, and each of those sites has a specific role that it plays in allowing this mRNA strand to kind of like move through and to create our polypeptide chain. So what do you need to know? A site, which think A comes first in the alphabet, so it's going to come first here, is going to hold your tRNA molecule. Your P site is going to be what kind of like holds your growing polypeptide chain. It's going to link them together, link together your amino acids. And then your E site, I like to think of as your exit site. E is for exit. It's where you're going to like release your tRNA molecule, because now you have built your polypeptide and so it can go on and be reused. You'll notice as we're building this polypeptide chain, right, that we're going to add some energy in order to form these bonds. We have to use energy along this process. But really, initiation is just this first bonding of mRNA to my ribosome at my start codon of AUG, my start codon on my mRNA. We were just talking about mRNA and the start codon, right, of AUG. Well, how do we know what amino acid that mRNA is coding for? Well, we use something called a codon chart. Okay, if we think back to the beginning of the year, there are 20 naturally occurring amino acids, and we use a code of 3 nucleotides on our mRNA sequence to code for 1 amino acid. So codon charts can look differently. They can look like this. I'll show you another one in a moment here. But you need to be able to kind of use them because they're really helpful, and they'll tell you what amino acid is coded for on what again? The mRNA. Okay, so on our mRNA, if my first letter is represented here, G, A, C, or U, my second letter, then I'm going to match it up here, U, C, A, or G, and then my third letter is over here. So let's just say very simply I have the code AUG, right? My start codon. I told you your start codon is always methionine. My first letter is A, my second letter is U, and my last one is G. Okay, so where do all those match up? You'll notice it's right here. It says AUG, and then it says MET. That's just the 3-letter abbreviation for the amino acid methionine, because sometimes they're long to say, right? And you'll notice it is the only one on here for methionine, right? That's important because that's how we know where to start translation, right? It's always going to start with methionine. So you only want there to be one of those because you only want it to start in one place. However, when we look at the others of these, let's say we had CGA, okay? CGA, arginine, okay? ARG. But look at all these other triplets also code for the same amino acid, arginine. There's even some down here, right? Well, is this good or bad, you might be saying, right? Why is this good? We notice that there's redundancy, right? There's many codes for just one amino acid. And that's kind of actually a good thing. You'll notice oftentimes it's the third nucleotide that can be very different, right? But still code for the same amino acid. Well, that can be helpful sometimes in mutations that occur so that we still code for the correct amino acid, even if there is some mutation along the way, and overall it sort of ends up all right. So that redundancy is actually a good thing in making sure we code for correct amino acids along the way. The last thing I want to point out is our stop codons. Our stop codons, you'll notice, do not code for an amino acid. Rather, they terminate the translation process. So there are 3 of them: UAA, UAG, and UGA are your stop codons. 3 of them, a little bit more variety, but again, not a ton, because it would be bad to stop your amino acid sequence before it was done. You wouldn't have a completed polypeptide chain, which would influence your protein. And remember, proteins help us function, so that would not be good, right? So, what do you need to know? Do you need to memorize this chart? No. Do you need to be able to use it? Yes. Will they give it to you if you need to use it? Yes. And should you have an idea what the three-letter abbreviations mean? Yeah, you probably should too. Okay, so these are your mRNA codons and your codon chart. Codon charts can look different. Sometimes they're in a circle. This is your first base. This is your second base. This is your third base. And then this is the amino acid that it codes for, or the stop codon, whatever it might be. So either of them you should be comfortable using. I also included here the three-letter amino acid abbreviations, so their full name, just so you know what they are. And yeah, you should probably be familiar with both of them.